Hi everyone, I'm Jayatri, Chief Bioscientist at the Franklin Institute, and welcome back to today's conversation about some of the science going on behind the COVID pandemic that we're all dealing with. Over the last couple of days, we've talked about some of the drugs that are being developed for the virus, some of the vaccines that are in development, and both of those were really founded on the underlying information that's provided by the genetic sequence of the virus. And so today I wanted to dig in a little bit deeper into that genetic sequence and the clues that it can give us about where the virus came from, how it works and why it's so infectious, and where we can predict it to go in the future. I'm an evolutionary biologist by training, so this kind of stuff is really interesting to me. But it's also another tool that scientists are using to predict the big picture of how this virus fits into what we know about viruses in general and what we know about the rate of evolution to help us plan for the future. So let's start with a little bit of basic information about how evolution works at the genetic level. So biological information is coded in DNA and RNA. So whether it's the information that our cells use to build us, or plants, or bacteria, or viruses. It's all DNA and RNA sequences. Now, as this genetic information is passed down from one generation to the next, there are random mutations that happen. A mutation can be any kind of a change in that genetic sequence. Most of these mutations have no effect. We call them neutral mutations. Some of them are mutations that make the organism less effective at reproducing, so those usually get deleted out of the population because they just don't reproduce and survive. Every so often, there's a mutation that actually helps the organism reproduce more efficiently or in greater numbers. And those kinds of mutations, because they're carried on to more offspring, they tend to increase in frequency over time. Now, what we can see is that where you see mutations increasing over time, they're likely to correspond to some kind of function. So that goes both ways. And by looking for those kinds of mutations and then trying to understand what purpose they serve in the molecule that they're encoding, they can give us some clues as to why those mutations are important and how they're helping the organism survive. So let's think about what that means when it comes to the coronavirus that causes COVID-19. So the sequence for the whole virus was published back in January by Chinese scientists who then shared the sequence with the world so that scientists all over could start to analyze the sequence. And what they found was that the closest relative of this particular virus was actually found in bats. And so we do think that this particular virus, like the SARS virus before it, came from bats, but has undergone some changes along the way. Now, it is 96% identical to the bat virus. It's about 80% identical, when you look at the sequence, to the SARS virus. So both of these are kind of interesting comparison points to look at to see where things have changed. Now, when you look at this new human version of the coronavirus, one really interesting change that scientists found was in the part of the spike protein that actually binds to the human ACE2 receptor. So we've been talking about this interaction between the spike protein on the virus. These are proteins that stick out of the surface of the virus, with the ACE2 receptor that's found in cells in our respiratory system. This lock and key mechanism is really the linchpin of how the virus is able to infect our cells. Now, when you look at the part of the genetic code that codes for the part of the spike protein that actually fits into the ACE2 receptor, you find some key differences that exist in this SARS-CoV-2 coronavirus that we're dealing with now that didn't exist in some of in its related viruses. And so that gives us a clue about why it might be so infectious. Because it turns out that these changes somehow, and we don't entirely understand how yet, make that interaction between the protein and the receptor a little bit stickier. So it's more efficient at infecting a cell. So that's one key difference that scientists are really interested in now testing, why those changes at the genetic level might cause changes at the functional level to increase how infectious the virus is. Okay, so we've got those changes. The second change that scientists saw when they were analyzing the sequence was an interesting site that looks like it's a place where a very common human enzyme can come in and cut the spike protein in half. So here's why that's important. The spike protein is actually divided into two parts. One part of the spike protein is responsible for attaching to that ACE2 receptor. The second part of the protein is actually responsible for helping the virus to then fuse with the human cell, and then the genetic information can then transfer. So what happens, the scientists think, is that with this new insertion of this chopping site, that helps human enzymes come in, cut off the part of that binds to the receptor once that binding has already happened, and that opens up that fusion site to make the fusion of the virus with the cell much more efficient. So that could be another reason why we think that this SARS-CoV-2 virus is more infectious than some of the other viruses that don't have this ability to just chop off and get rid of the binding site to then get moving with the fusion and the infection. So what scientists think so far is that based on the sequence comparison we've seen, that first change of tweaking the actual binding site might have happened while the virus was still in animals. It looks like the change with the addition of the cut site might have happened after it migrated into humans. So there are some other analyses that we can do to test in more animals and compare more sequences to start to tell the difference between these two hypotheses. And that's going to be helpful for us to understand how viruses move back and forth between animals and humans and how they're evolving within humans as well. Now, the big question is, how is a virus going to continue to change as we look into the future? And this is important as we're thinking about developing vaccines for the virus, because if we're to develop a vaccine that targets a particular site on the virus, but then that genetic sequence changes, then that might render any vaccine no longer effective. So we need to be able to understand what might happen to be able to prepare for that eventuality. What we know so far, remember that most mutations that occur are generally neutral. So far, looking at how fast this particular virus seems to be collecting mutations and comparing it to something like the flu virus, it looks like the flu virus is 2 to 3 times faster at accumulating mutations than this coronavirus. And so we're hopeful that we're not going to see this virus changing as fast as we see with the flu virus. So compared to other similar coronaviruses, we might expect that drastic of a change to take, maybe a year or 2, maybe 3. But it's still unpredictable. So what scientists are doing are trying to collect samples of the virus from all over the world and sequencing them. There's a really fascinating website called nextstrain.org, where scientists are depositing all of these sequences and looking at the relationship between each other to track how fast the sequence is changing, where it's changing, and what that means. So as these sequences continue to collect, we'll have a better idea, again, of how fast these changes are occurring, where they're located in the protein, because again, mutations in some places might not matter, while mutations in other parts of the protein might make a big difference. So I hope that helps you understand how analyzing genetic information in a theoretical way can both help generate hypotheses that we can then experiment with in the lab to understand how the virus works a little bit better. And then we can also start to predict how the virus might change in the future to prepare accordingly. Thanks again for joining us today. We'll be back again tomorrow talking some more science.